Hello, my name is Mr. Neumyer,
And my name is Emmy Smith.
and today we'll be talking to you about botulinum toxin, one of the causes of food poisoning.
To understand the toxin, which I'll be referring to as BTX, we first need to know how neurons transfer signals. Looking at this picture, see the large yellow blob on the left called the soma with the small strands called dendrites. Also, notice the strand in the middle called the axon. The axon connects to the soma on one end and splits off on the other end into multiple strands called synapses. Neurons create a network by connecting their synapses to the dendrites of other neurons. A signal starts in the dendrites, travels down the axon, and then jumps to the next neuron. However, the signal doesn't actually travel through the synapse to the dendrite of the next neuron. When it reaches the synapse, the signal triggers the release of neurotransmitters, which are little chemical signals, into a gap called the synaptic cleft, and the signal ends. This is the presynaptic neuron, this is the dendrite, and right here in the middle is the synaptic cleft. The neurotransmitter traverse the cleft, they attach to specific receptors on the dendrite, and a new signal is started in that neuron. When a signal reaches the synapse, it activates a set of proteins called snare proteins that fuse a vesicle that contains neurotransmitters to the membrane of the neuron. Some examples of snare proteins are syntaxin in red, SNAP25 in green, and synaptobrevin in blue. You don't have to memorize the names of them though, just thought it would be pretty cool to share with you. These snare proteins allow the neurotransmitters to enter the synaptic cleft and bind to their receptors. Well, just like it always has before, nature found a way to screw this up. The cool-looking protein here is the focus of this presentation, botulinum toxin. It is produced by the bacteria of the Clostridium genus, which are found all throughout homes and the outdoors. That's kind of scary. The term botulinum toxin actually encapsulates a group of toxins, which are referred to by their serotypes. Serotypes differ based on their surface antigens. We are focusing on serotype A in this presentation. The spinning model next to the picture is a 3D model of BTX. The pink areas are the alpha helices, the yellow strands are the beta pleated sheets, and the tiny purple bits are the 310 helices.
When introduced into an organism, botulinum toxin cleaves SNAP25, one of the snare proteins responsible for vesicle fusion in the neurons. And then, because the vesicle cannot be fused, the neurotransmitters are not released, so the signal cannot be continued onto other neurons.
BTX is able to do this by attaching to SNAP25 using the active sites located on the right side of the image in the middle of the screen. The picture of the JMOL model shown to the right is a close-up on BTX with focus on the bulge with the active sites. The active sites are painted green in the model. These active sites are lysine, found at amino acid 41, isoleucine, found at amino acid 115, valine, found at amino acid 129, and cysteine, found at amino acid 134.
Here's an important life lesson to take away from this presentation. Always clean canned meats. The bacteria that produces botulinum toxin thrives on improperly sterilized canned meats. Botulinum toxin also thrives on poorly sanitized areas such as counters, flooring, carpets, soil, and dust. The ingestion of Clostridium spores through these means is commonly referred to as food poisoning. When exposed to an anaerobic environment, the spores germinate and produce botulinum toxin inside the body. This can be fatal if not treated immediately because it can paralyze the person's lungs and may prevent them from being able to breathe. What's awesome about this protein is that scientists have found uses for botulinum toxin in a variety of surgeries. When injected, it can be directed to only paralyze a certain area. You've probably heard of Botox, a cosmetic treatment to improve beauty through paralysis. Botulinum toxin can also be used for eye realignment surgery, where it paralyzes the muscles that move the eyeball, so the eyeball can be rotated to look in the correct direction. It is also used to treat people with muscle spasms because it paralyzes their muscles. Another interesting application for botulinum toxin is with migraine reduction. Migraines are caused by pain signals being sent through neurons in the brain, and botulinum toxin can stop those pain signals from being sent. Since the pain is perceived through the neurons, it can reduce the pain in certain cases. Migraines are an area that researchers are looking into using botulinum toxin for because of its interaction with neurons. Through a few studies, researchers have found that botulinum toxin in fact does reduce the pain felt from migraines and reduces the total number of migraines.
We took the JMOL model shown previously of the bulge with the active sites and 3D printed it. The model fits in your hand. It isn't large. It was formed using black material, so we couldn't use the same color scheme as before because some of the colors didn't look as nice as they did on white. We painted it with oil-based paints because those stood out the most and wouldn't wash off as easily. The green paint indicates where the alpha helices are located, but a few couldn't be colored due to their location on the inside of the model. The white paint indicates where the active sites are. Notice that they aren't as accessible as one might imagine. This allows the protein to be more specific. The beta-plated sheets are not painted because many are located in hard-to-reach places in the model. Thank you for watching our presentation. We hoped you enjoyed it and learned a lot. If you have any suggestions or questions, send me an email at the address acneumyer@gmail.com. If you have any complaints, send us an email at the address emmyhsmith@gmail.com. Thanks, and have a good day.